One of the first steps in many data science projects is exploring and visualizing the data. The pair plot is a common and powerful visualization tool to explore patterns in the data, identify potential problems with the data like outliers and mislabeled points, and gain insights that might help you decide which methods to choose. In this video, we're going to go through how to make and interpret a pair plot and some of its strengths and weaknesses. To start, we're going to import seaborn as SNS, and seaborn is a great tool for making nice, quality visualizations with very few lines of code. We'll use the penguins dataset. We can load this from seaborn, and let's take a look at the dataset. We can see there are several columns. The first is the species, and that is the target. This is the value that we're going to try to predict. And there are four numerical features that we're going to look at: the bill length, the bill depth, the flipper length, and the body mass. And we're going to try to use these four features to predict the species. And let's take a quick look at the species that we have to work with. We can see there are three different species: the Adelie, the chinstrap, and the Gentoo penguins that we're going to try to predict based on these four numerical features. The pair plot is a great tool to try to understand the relationships between these four features and also how they can relate to the species, the ultimate target. And we can make our pair plot just with one line of code, sns.pairplot. We tell it the data set we're working with, and we'll also give it a hue for species, and that'll just tell it different colors for the different species, which is what we're trying to predict. Here's our pair plot. There are 4 rows and 4 columns, each corresponding to one of those 4 features. And we also have 3 different color points that we're looking at here, and each of the colors corresponds to a different species. In each of these scatter plots, we see one of those features plotted against another. So right here in the lower left-hand corner, we can see the body mass plotted against the bill length. And every scatter plot in that bottom row has the body mass on the vertical axis. And similarly, every scatter plot on the second to last row has the flipper length on the vertical axis. If we look vertically, we can see that every scatter plot in the first column has bill length on the x-axis and this continues throughout the whole plot. Now, based on this, you might be wondering, so what goes on the diagonal elements? Because that would be, in this first element, the bill length versus the bill length, which should just give you a straight line. It doesn't provide that much information. So instead, they give you this marginal density plot, which tells you something about that individual variable. So in this case, the horizontal axis still has those same values, the bill length in this case. But on the vertical axis, you can see the distribution of points that fall within this particular range. So for example, in the case of the bill length, you can see that the Adelie penguin seems to have the smallest bill length of the 3 types, and the Gentoo and chinstrap penguins seem to have overlapping bill lengths. One thing you might notice is that this pair plot is actually repeating itself for some of the values. So for example, if you're plotting bill depth versus bill length, that's going to be the same information plotted on the other side of the diagonal for bill length versus bill depth. When looking at this pair plot, we can start thinking about how we can use this information to see if it's easy to separate out these 3 penguin species from one another based on these variables. And one thing that stands out here is that, for example, we can separate out all of the green Gentoo penguins from the other 2 penguins using body mass versus bill depth. These seem to be linearly separable. However, if we were to use those same 2 variables trying to separate out the blue and the orange penguins, we really wouldn't be able to see any difference. However, looking at bill length, for example, we do get a clearer differentiation between the blue and yellow penguins. Another thing we can learn from this plot is that it seems that the body mass is linearly correlated with the flipper length, which might make sense. If you have larger flippers, maybe you have a larger body mass, or you might need a larger body mass to support larger flippers. So that was a relatively simple example with just a few features that are relatively separable. But let's look at something a little bit more complicated. Now we'll take a breast cancer data set from sklearn. Let's load in the data as a data frame and print out the columns. There are a lot of columns here with many different features, so let's pull up the website to see what this all means. If we pull up the website that corresponds to this data set, we can see that there are 30 numeric predictive features that will predict whether these cell nuclei are either malignant or benign. And these include variables like the radius, the texture, and several other variables. And it includes these 10 different attributes with their mean, standard error, and their worst or largest of the values that were measured. So this is a lot of data, and it's going to be a lot to parse through with a pair plot by itself. We can't really plot a 30 by 30 pair plot. but we can still use a pair plot to try to learn a little bit about how these variables are related to one another. And so one hypothesis I have right from the beginning is looking at these variables, the mean radius, mean perimeter, and mean area. I would hypothesize that these three variables are correlated with one another, and that's a great application for a pair plot. Let's give this a try by plotting the first four variables, and we're also going to add in the target data so that we can color and figure out what's the difference between the malignant and the benign tumor cells. When we take a look at this data, we do see a very clear linear relationship between the perimeter and the radius, which makes a lot of sense. And we also see what appears to be a quadratic relationship between the area and the radius. Again, that makes sense based on our understanding of what an area is. Interestingly, we don't see much relationship between the texture and any of these three variables, which means that these must not be correlated with one another. Another thing we can learn from the data is this distinction between the orange and blue points. So the blue points correspond to the malignant cells, and the orange points correspond to the benign tumor cells. And one thing we can learn from this simple plot is that the malignant cells tend to be much larger, have larger nuclei than the benign cells. This seems like it'll be an important indicator if we're trying to use this information to predict whether a cell is malignant or benign. And in fact, if we just use these data, we're probably going to be pretty good at predicting whether a cell is malignant or benign. If we have some other hypotheses about some of the other features that we have to work with, we can just go in and add individual features that we think are interesting to a new pair plot, plot it, and then look for any relationships or any interesting features from these data. The pair plot is a great tool for plotting numerical data and learning about the relationships between features. It's best for datasets with between 3 and 6 features. As we saw with the breast cancer dataset, using many features in a pair plot can be difficult to understand and overwhelming, but a pair plot can still be interesting if you can select a few key features of interest. Another weakness of the pair plot is that you can't learn about more complex relationships among 3 or more variables. Dimensionality reduction techniques like principal component analysis can help you understand higher dimensional datasets. I hope you enjoyed this video today. Thanks for watching.